All right, so we are back here to the laboratory, and after what you saw in the field, we were showing all the different accessions of Jatropha, reminding you that Jatropha is a biofuel plant. So the reason we are doing all that work and trying to do selections and improvement is because of this. Jatropha produces the seeds that have an excellent oil content. The quality of the oil is excellent for biodiesel production. And this is a sample of Jatropha oil, is pure Jatropha oil extracted from the seeds. This oil has excellent characteristics for being used as biodiesel or even jet fuels. So what we are doing in the field is towards the improvement of this plant so we can have increased yields, a plant that will be productive here in Florida. But here in the lab, we're doing also 3 different projects that address those issues in a different manner, which is more using biotechnology tools. The first one is that once we have our selections in the field and we want to multiply them, we need to be able to multiply large amounts of plants, make them available to growers. So the best way of doing this is using in vitro clonal propagation. So we're using tissue culture techniques to create clones of Jatropha that will be the superior plants that we'll be selecting in the field, and therefore those plants will have high yields, high productivity, maybe cold tolerance, and some other characteristics of interest. The other thing is, because we have all that variability that you saw in the field, we need to find a means of identifying if there are some genes related to that variability. So we are extracting DNA from the leaves of those plants, each accession that we have in the field, and we are running DNA profiles to compare them and see how closely related they are or how genetically variable they are. Genetic variability is something desired because the more variability, the more genes that we can introduce into new crosses and develop new cultivars. And finally, the third project is a project that we have in collaboration with NASA. Since 2007, we have been working with NASA on some space experiments, and NASA became particularly interested in Jatropha because of the potential for biofuel use. So we have already sent 5 experiments to the International Space Station National Laboratory using Jatropha cells. And the idea is that we know that microgravity will induce some changes in the gene expression of plants. So if we can expose those cells to microgravity, have a differential gene expression in space, now we bring those plants back, regenerate them into little plants, put them in the field, maybe some of the genes that are activated by the effect of microgravity can help accelerate the breeding and the genetic improvement process in Jatropha, which will save us maybe 10 years of work. So these are basically the 3 projects that we are carrying here in the lab. We have a biological assistants working with us, graduate students, and a visiting scientist as well. So it's a whole team effort towards the development of these new Jatropha cultivars. And we count also with the sponsorship not only of support of NASA, but the sponsorship of This Energy, which is a private company.